cellular respiration begins with glycolysis. So when we look at this word, glycolysis literally means the breaking down of sugar. Lysis is breaking down, glyco is sugar. We know that cellular respiration is the idea of breaking down sugar to get energy the cell can use. So we want to kind of figure out what exactly and who. And so when we start with the who, this is actually something that all organisms and really all cells perform. All cells perform respiration. Respiration is really going to come in 4 main stages, but all organisms will use glycolysis. So even bacteria will use glycolysis. We know that the electron transport chain is in the mitochondria. Not all organisms have a mitochondria. So focusing on the idea that this is actually a conserved or saved or repeated process. It happens in all organisms. And so we really want to focus on the idea that this is actually going to happen in the cytoplasm of all cells. And what's actually going to happen is that we're going to break a 6-carbon sugar into 2 3-carbon sugars. That's the main idea of glycolysis. We're actually going to break a 6-carbon sugar into 2 3-carbon sugars. Well, then the question is, why does this happen? Because we are actually trying to transfer the electrons. We're trying to take advantage of the electrons that are in there. So the main idea of the why is that as we slowly break down this carbon compound, we're going to be able to transfer some of the electrons' energy. And we're going to transfer them to a carrier. So for example, NAD+ is going to combine with 2 electrons, 1 hydrogen ion, and become NADH. We actually see the oxidation of sugar. So we're oxidizing sugar, oxidize glucose, reduce the carrier. So we're gaining electrons here, we're being reduced. The sugar is going to be oxidized. And this is going to happen when we need a source of energy. So when we need ATP, we're actually going to begin this process. This is just one step in the process, or one stage, really, because it's actually about 10 steps. So 10 steps, one stage, trying to get ATP. And here's the little diagram of ATP, reminding us that the energy is in these phosphate bonds, that we're actually going to go through many, many steps to build this ATP, but keeping in mind the 3 phosphates have that high energy. So breaking down sugar, all organisms breaking down sugar to transfer the energy to a different source. So next we look at the actual series of steps. So glycolysis occurs in about 10 enzyme-catalyzed steps. Thankfully, we don't have to know every step. We don't have to know exactly what happens in every step. We're going to focus kind of in on some smaller series of steps. So we start with glucose, which is a 6-carbon compound. So this is where we start. And we'll notice that we end with pyruvate. Pyruvate is a 3-carbon compound, but we're actually going to end with 2 pyruvates. So we start with a 6-carbon compound and we break down into 2 3-carbon compounds. You're not going to see carbon released anywhere, so no release of carbon dioxide. What we see in the first set of steps, so if we kind of divide and look at steps 1 through 3, steps 1 through 3 are known as an energy investment period. Because what we actually see is that we use ATP and we use ATP. So we have invested or used 2 ATPs. That's kind of unexpected. We're trying to make ATP. Why are we using it? It turns out that the investment of these 2 ATPs allows us to change the shape to secure glucose. Keep glucose in the cell. So we know that glucose is too big to diffuse across the membrane, but we do have a transporter for it. We do have a protein that will transport glucose in and out. And so what we see is that we actually want to change the shape of glucose so that it can't exit the cell. So we invest ATP and we end up with a pretty different shape. And this now can't bond to the transport protein, so therefore it can't move, it can't leave. And what we're going to focus on is the second time this happens. This enzyme, phosphofructokinase, is what we call a commitment step. Once we have invested this second ATP, we need to go ahead and recoup some of that ATP. So this step is only going to happen if we really need the rest of the reaction to happen. So we'll see in a little while that we're actually going to have an inhibitor that comes into play to inhibit this enzyme later. So first 3 steps is just the idea that we're actually going to invest energy. And then we keep seeing this idea of isomerase. And isomers are the same formula different shape. Same formula, different shape. So what we see is that we're still a 6-carbon compound. We've just changed the shape a little bit and added these phosphate groups onto the end. The next step is kind of a big step, and this is where we split the 6-carbon compound into 2 3-carbons. You'll notice these 2 3-carbons aren't quite the same, so there is this other little step here that changes dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. You don't need to know these names. You do need to know G3P, this one. G3P. G3P is a 3-carbon compound that we end up with 2 of them. After we do this isomerizing, we change the shape around a little bit. We end up with 2 G3Ps, which we'll see come into play a little bit later. So these 2 G3Ps then actually get an electron removed. So these are actually what get oxidized. NAD plus gets reduced. So right here we actually see the creation of 1 NADH per G3P, or 3-carbon compound. And because this idea, so this is per, because we have 2 G3Ps, we actually have 2 NADHs. So because I have 1 NADH per G3P, 2 G3Ps, what I'm getting out of this is really 2 NADHs, because it's happening for both of these molecules. We see that we actually get an ATP out and an ATP out, but keeping in mind, if that's 2 ATP per G3P, that's actually 4 ATP. We do need to take a second and talk about this formation of ATP. This formation of ATP is called substrate level phosphorylation. So big term here, substrate level phosphorylation, meaning that I add a phosphate group on, phosphorylation, and it happens directly from one substrate to another, versus oxidative phosphorylation, which we'll see later. So what's going on here? I start with a 6-carbon compound. I invest some ATP to change the shape so that I have a slightly different shape. It can't leave the cell. I commit once I go through step 3 with this enzyme phosphofructokinase. Then I split into 2 3-carbon compounds, each of which makes 1 NADH, 2 ATP. 2 G3Ps really gives me 2 NADHs. 2 G3Ps really gives me 4 ATP. So I've invested some ATP, I've produced some ATP, and I've definitely produced 2 electron carriers. And here we have our 2 3-carbon compounds. So we're going to look at the next part of the notes. It's another summary of what's going on. So what is the main idea of glycolysis? We do want to keep in mind all cells perform glycolysis in the cytosol or cytoplasm. We take a 6-carbon glucose and break it into 2 3-carbon pyruvates. So we haven't lost any carbon. We have a net gain of 2 ATP and 2 NADHs. 1 per pyruvate. If we look here, we had our energy investment where we invested 2 ATPs into the glucose. We ended up paying out 4 ATPs total, 2 NADHs and 2 pyruvates. So our net, once we've talked about what we invested and what we got out of it, our net is that we get 2 pyruvates, 2 ATPs, and 2 NADHs. We're not really going to worry about the water right now. We're going to focus on the carbon, the ATP, and the NADH. So it turns out that this reaction will actually happen at different speeds depending on how much ATP the cell needs. So we're going to talk real quickly about how to regulate this. Why would we want to regulate this? We do not want to go through extra steps if we don't need ATP. If we don't need ATP, we don't want to do all these reactions. So how does the cell know if it needs ATP? And this is where phosphofructokinase really comes into play. Phosphofructokinase is an enzyme that is allosterically inhibited by ATP. So, ATP binds to an allosteric site on phosphofructokinase. And it inhibits that enzyme. So, if I have a high concentration of ATP, this reaction will not happen. So if my cell already has plenty of ATP, I don't need to keep going with all these steps. I'm just going to shut the reaction down. I'm not going to use any in this step, and I'm not going to keep making ATP that I don't need. So that's a huge regulatory step, the fact that there's this enzyme at the very beginning almost that can be inhibited if I have plenty of ATP. The other thing that we'll actually see a little bit of that I want to mention really quickly is this idea of G3P. Right? We saw that the 6-carbon breaks into 2 3-carbons. G3P is actually used elsewhere in the cell. So this is another regulatory step. If I have plenty of ATP, I don't keep using G3P. actually can use it elsewhere in the cell to build other things like a carbohydrate chain on my cell membrane. So I'm not going to really use